paper today is entitled Breast Cancer Stem Cell Antigens as Target for Immunotherapy. I will start by introducing the term immunotherapy. Okay, just before I begin, I want to say that I didn't mention a lot of other papers. Like I just focused on this paper. I didn't get many references. Okay, so that's just this one. So immunotherapy, it is a treatment by using the body's own immune system to fight off cancer. Now, there is a lot of obstacles that face that is faced by the immune system when targeting cancer cells. One of them is it might not be strong enough to kill all the cancer cells. Cancer cells can produce signals that stop the immune system from attacking it, like they are evading the immune system, if you want. And cancer cells hide or escape from the immune system. Now, we have 4 types of immunotherapy, as I know of. One of them is monoclonal antibodies. They can trigger and help the immune system to fight. We have checkpoint inhibitors, vaccines, and CAR T cell therapy, which is chimeric antigen receptor T cell. Okay, these therapies are all used nowadays, and we are currently there is a lot of clinical studies that are conducted using these therapies to target cancer, all types of cancers. So why targeting CSC is so important? If you remember, I talked about last time what is CSC, cancer stem cells, and we defined it and why they are so important and crucial for the treatment of cancer from its roots. So, like I said last time, they play a key role in tumor development, dissemination, and resistance to therapy. So we can say that they are the root for cancer. They are the cause why cancer can metastasize and recur, and they are the problem behind the not successful therapies. So it is crucial to identify suitable molecular targets expressed specifically by CSCs, which are called oncoantigens, as effective anti-cancer vaccination targets. So this is the goal of immunotherapy these days. They want to target specific antigens found on cancer stem cells, not on the tumor body cells. I mentioned before last time that there is a controversy over this hypothesis because it's relatively new. They are still working on approving it worldwide. But some of the problems with this cancer stem cell hypothesis is the lack of universally defined cell surface markers. So they cannot define a specific marker that is found in all types of cancer, or at least the same histotype, or in different patients. And the high heterogeneity among various tumor types and also among tumors of the same histotype from different patients, as I said. Now, they mentioned in the first part of the paper some statistics. I talked last time also about statistics. I did not mention them in my presentation this time. So they also said that breast cancer accounts for about 12% of all new cancer diagnoses in 2018. I said that last time. And I want to add something that they wrote in this paper. I found it interesting that there is a type of cancer, breast cancer, called that they say HER2+, which is a protein that helps breast cancer cells grow quickly. HER2 is human epidermal growth factor receptor 2. So if this receptor is present, they say that it can have the highest rates of early cancer recurrence and metastasis. So they thought that targeting this specific receptor can stop recurrence or metastasis. And they also really focus on triple negative breast cancer. So it is estrogen receptor negative. They don't have estrogen receptors, and progesterone receptor negative. And this receptor I talked about now, come in, also it's not present. This is what triple negative means. So if breast cancer is said to be triple negative, it can grow quickly, is more likely to have spread at the time it's found, and is more likely to come back after treatment. So they are really targeting these two types of cancer right now. Now, in the paper, they mentioned cancer stem cells immunogenicity. So as I said, cancer stem cells can evade the immune system with a lot of different ways. They also found that cancer stem cells bear a lower susceptibility to immune cell killing than differentiated cancer cells. So this is part of why cancer stem cells are really hard to kill, and they are present in all types of cancer. Now, in spite of this statement, different approaches were used to find a feasible treatment. They started using cancer stem cell immunotargeting using vaccines. As I said, they want to target specific receptors on cancer stem cells. They are using CAR T cell therapy and other different therapeutic approaches to target specific stem cell markers. So I'm gonna start by talking about the anti-CSC vaccination. Okay, they mentioned in this paper some clinical or preclinical trials, ex vivo and in vivo, on mice. They found some successful results. Okay, they used dendritic cells to induce anti-CSC specific humoral and cellular immune response. So it is a way to make a vaccine using dendritic cells. They were successful in eliciting measurable and specific anti-tumor immune responses, as I said, but in mice, not in humans. They are still in preclinical trials. And this was a good approach because it did not induce strong side effects. But the one drawback they really found is that it's cost and time consuming and patient specific treatment. So they have to study every patient by himself or herself and target specific receptors on his specific cancer. So like I said, cancer is very heterogeneous in the same patient. It can be in the same patient and in a lot of different patients. So it is really cost consuming and time consuming. They are trying to standardize it like other treatments. The second approach they used is CAR T cell therapy. It's also to target specific cancer stem cell antigens shared by tumor histotypes and patients. So it is more specific than dendritic cells vaccines. I talked about them earlier. It's not targeting the whole antigen repertoire on these stem cells. Some promising results were found also in preclinical trials in mice. And the one drawback was it was expensive, and they found that it has some serious side effects, including cytokine release syndrome. So I think vaccination using the traditional way or using dendritic cells was better than using this CAR T cell therapy. Okay, so now we're gonna delve into the core of this paper, which is breast cancer stem cell antigens identification. So I talked about cancer stem cells in general. Now I'm gonna focus on breast cancer a little bit, as they focused in this paper. So they included this figure. This is a procedure they devised to find a way to find the specific markers found in any cancer stem cell. So it is a lot of steps. Okay, I try to understand some of them, but they are really advanced. Just to mention, they are using this method to be able to identify new or novel potential targets, mainly for diagnostic breast cancer stem cell detection, for immunotherapy, and for pharmacological treatments. So by using this specific procedure, they found some new antigens they can target using immunotherapy. I will talk about them in a few. The first one is called Crypto1. It plays an important role in cancer progression. It is a class of antigens. Like I said, it is important in cancer progression. It is expressed in HER2 plus and triple negative breast cancer cells, stem cells. It regulates self-renewal, migration, and EMT of these cancer stem cells. Now, what is EMT? EMT is epithelial to mesenchymal transition. It is a thing that differentiated cancer cells do to start back as stem cells. So this is an important phenomenon that the differentiated cancer cells do to evade the immune system. So they tried and thought that this is a good target for immunotherapy. They devised a vaccine with a DNA plasmid that codes for the full-length Crypto1 oncoantigen, and studies found that this vaccine decreased lung metastasis and stops tumor growth in mice. So every clinical trial mentioned in this paper was really done in preclinical trials in mice. So there is nothing definitive and nothing efficient to say that. But they are getting somewhere. They are really trying to discover some of the specific markers on these cancer stem cells. Now, the second target mentioned in this paper was TNM4, which is tannurine transmembrane protein 4. It is involved in the development of the central nervous system and the organogenesis of additional tissues. Now, why it is important in cancer? Because the dysregulation of this protein can contribute to human carcinogenesis. So they thought that this can be a suitable immunotherapy target. A third antigen is called SCARA5, which is scavenger receptor class A member 5. It regulates the import of L-ferritin into cells, contributing to iron uptake. It is important in breast cancer stem cell maintenance. So all these receptors or things I'm mentioning, they are important in the progression of the cancer in breast tissue. And this one was not an ideal target for immunotherapy because mainly it was constitutively expressed by other epithelial normal cells in the body. So they cannot target this cell, and they could kill other normal cells in the body, which are present in the gastrointestinal tract, gallbladder, testes, and hepatocytes. So they also find a fourth one, which is called TLR2, toll-like receptor 2. It's a type 1 transmembrane protein. It can induce production of IL-6, TGF, beta, and VEGF, and by this they can activate other pathways to promote BCSC survival, proliferation, EMT, and invasion. They found that this was a good this was actually a good BCSC target for immunotherapy, because as they found in studies on mouse models, that the TLR2 inhibition strongly decreases tumor growth and metastasis in mouse models. So one last receptor they talked about in this paper is XCT. They mentioned this receptor or marker specifically because it regulates the import of cysteine in exchange with glutamate, which is contributing to the synthesis of antioxidant molecule glutathione. So this is a way cancer stem cells, by using this antigen, they can evade the immune system by using antioxidant molecules. So they thought that we can target this marker so they can stop producing antioxidants and they will die by themselves, so they found that this was a good target for immunotherapy. It could be used as a therapeutic target for pharmacological inhibition other than immunotherapy. It was good in using drugs. And some of the drugs mentioned that was used targeting these antigens are called Erastin, SAS, and sulfasalazine, or sorafenib. So these drugs, really with weird names, was mentioned in this review, but they are actually not very effective in humans because they are insoluble under physiological conditions and have poor metabolic stability. So they thought that immunotherapy was better than pharmacological treatment. And they found also that XCT vaccination can be combined with chemotherapy, regular chemotherapy, to reduce metastatic disease. They mentioned a study and they found that treating patients with XCT immunotherapy and chemotherapy reduced this metastatic disease and stopped CSC self-renewal and survival by increasing cell sensitivity to chemotherapeutic drugs. So it was a really good combination, and I hope they try and combine from other treatments together with surgery. So to conclude, targeting CSC with innovative immunotherapy is of utmost importance for the goal of curing cancer from its roots. Cancer stem cells are found to be the cause behind metastatic cancer, so this is why they are targeted in new clinical trials. So to achieve tumor eradication, the ideal vaccination targets are those present on differentiated cancer cells and overexpressed in cancer stem cells. It was mentioned in this paper also. And for CAR T cell therapy, it is the identification and selection of appropriate CSC marker that is crucial for success.